Good afternoon, everyone. My name is Kimberly Perez, and I have the pleasure of introducing our speaker for this afternoon, Elena Ramos Elvira, a visiting scholar from the Universidad Complutense de Madrid. And you may know that we have an MOU that resulted from Dr. Hernandez's Fulbright. And thus far, we have sent about 3 students to Spain, and they have sent us 2 students to UGA or to the Hernandez Lab. So a little bit more background on Elena. Elena is a biologist who holds a master's degree in conservation biology from the Universidad Complutense de Madrid. She is currently a PhD student under the advisement of Dr. Jose Aguirre and is currently exploring microplastics in various systems, including the White Ibis. So welcome, Elena, and I will turn it over to you.
Thank you, Kim, and thank you all for coming. Well, as she just said, I come from Spain. I am at University Complutense of Madrid. And well, I'm gonna talk to you a little bit about what do we do in our group, our biodiversity monitoring group, and why is my research about. So to give you a little bit of settings, we are across the Atlantic in Spain, right at the center of the peninsula, it's Madrid. And here we have a Complutense University of Madrid. We have 2 campuses, but our main one is Moncloa campus, which is right beside the historic city of Madrid. You can see it here. This is the whole historic center of the city. In case any of you has been in Madrid, this is the Retiro Park, which we like it to compare to Central Park in New York, but we wish. And here is the whole campus, and it is in a really privileged location because it is surrounded by different natural areas as Monte del Pardo, Casa de Campo, which are they are conservated as to a point that El Pardo you cannot even enter, not because conservation policies, but because it belongs to the King of Spain. But that's another topic. It also, the Manzanares River flows throughout the city from north to southeast, and on the southeast part, you can find another regional park. So we are located in a really nice spot for biodiversity. More even when we think about migratory routes, if you look into how birds go from Europe to Africa, there are 3 main spots, 3 main areas: Iberian Peninsula, Italian Peninsula, and the Balkan Peninsula. So all birds that go through Spain, most of them, they go through the middle of the peninsula where Madrid is, and we could find lots of birds migrating from the north of Europe. So with all of these characteristics, some years ago, they tried to create a program to identify, evaluate, and manage the biodiversity that we have. So the biodiversity monitoring group was created, Grupo de Seguimiento de Biodiversidad in Spanish, and it is a standardized monitoring program. This implies that we want to have some clear objectives to try to achieve our goals. We really like to focus on continuity, since it is not the same, or it's not the same to just have a specific moment in time, or rather than having lots of years. And also we try to have a standardized methodology that any researcher in the future will take up. So a little bit of history. In 2010, 2 professors at the university created the fauna monitoring group. They started monitoring birds, butterflies, and ants. They wanted to try to bring nature closer to students, and that the students could practice and could learn some activities outside the classroom. So then they started having some other activities such as nets boxes, bird banding, butterfly transects. And afterwards, they were including some more as other monitoring, bats, micro mammals. And then in 2019, the plant team joined us, and we passed from fauna monitoring group to biodiversity monitoring group. I want to remark that even though we started in 2010, in 1995, bear banding station started, and it has not stopped since that moment of time. And in 1999, we started to have some monitoring in a white store colony, right in a close town to the university. So how do we manage this? There are lots of professors that they try to plan, supervise, and design this analysis, these activities. But of course, without the help of some volunteer students, we couldn't do all of this. We try to bring them in the activities, and they will collect data, they will help in the field, and of course, they will be learning new activities. And then once they have been with us for some period of time, they will become experienced students, and they will be able to manage data, to organize some other things. We also we really take pride in our transversal transmission of knowledge, which will mean that even though you may be a new student, if you are learning and if you are participating in these activities, you can learn pretty fast. And maybe you are an undergraduate, second year, third year undergraduate, and you could be teaching some new activities to a PhD student, for example. So we don't care so much about the rank that they hold, but rather than what experience they have. We separate in 3 layer groups: vertebrates, entomology, and plants. I am in the vertebrates section and in the ornithological unit. However, I will be spending a little bit of English. What do they do in entomology? Well, they do some transects along the campus. You can find lots of habitats in this transect. So they want to look at what kind of species there are, how they are distributed, their phenology, and of course, how the urban environment and how our human activities will affect them. So for example, these are some species that you could find in the campus, but also with this continuity that we want to have, you could see how the abundance of individuals have been growing depending on the occasion. For example, when the European Union banned the use of glyphosate, this abundance went up. And of course, with the COVID-19 situation, when everyone in Spain was confined and we couldn't be home during the spring, it was really remarkable. In the plants team, they are creating a digital plant catalog at the university. It can even be used for the whole city of Madrid, since the campus has different habitats. And we also try to geolocate and characterize these habitats. Some orchids pop up in the middle of kind of nowhere. This is like next to a road. So we are trying to have some micro reserves at the university. We are trying to talk with the government of the university. They are not listening a lot to us, but we try to do what we can. So this is a nice little spot where we try that students don't harm these orchids. Well, sometimes we achieve it. There are some other times where people just party and we cannot do anything about it, but we are trying. And also we have achieved that they don't mow path edges, which allow us to have lots of diversity. And of course, we are characterizing habitats at the university. About the vertebrates section, we do some micro-mammal surveys. We do it twice a year. We put some Sherman traps, and we try to catch micro-mammals. we're not very successful. We don't think we have a really populated area of micromammals, but still we try. And if we get some, we will, oops, sorry. We will put some ear tags on them. We also monitor bats. We have been doing for a long time these visits to these boxes where they could refuge. But in the last 2 years, we have some project going on where we do 3 visits per month. We do some transects, and with an app, we can see how their ultrasounds could let us know which species there are there. And Pipistrellus is the most abundant one. And our most participating activity, the one that I like the most, is scientific bird banding. For those of you who are not familiar with this, we catch some birds in mist nets, and we place a metal band on their leg. It has a unique alphanumeric code, and this way we could identify this individual for their whole life. We try to collect data. If we know, if we can get the age, the sex. We also look for the physical condition and some biometrical parameters. And of course, we collect samples. Since we already have the bird in our hand, we try to take the most out of it. And we collect blood samples and fecal samples. This is some examples of what we can find. We even collaborate with some faculty members here, such as Dr. Sonia Hernández or Dr. Michael Yapslik. And we also take some parameters as the wing length, the tarsus, some feathers, the weight, and depending on the project that we have in that moment, we will take some other samples. And we use the constant effort size, which in Europe is like really extended. We try to have some standardized method that anyone could relate to, and so that we can compare different bird banding stations. For example, we could see throughout the whole year if some avian communities have changed. For example, we can see how the bald-tip is a bird who we will have more on the winter months, and the tree sparrow and house sparrow, which are more generalist species, they could be found the whole year. So with these constant effort sites, we just place the nets in the same exact spots along the whole year, at least twice per month, and they will be placed there before sunrise, and we will be there for 5 hours. We mostly catch passing birds, but that's not always the case. For example, this summer we got some hawk. There's some other cases where we have got some nightjars or some little owls. So that you can see the diversity that we have on campus. You can see here, this is our faculty building, and these are one of our nets. We also, of course, do research. There are some undergraduate research projects that students have to do. So we help them with our data, we help them do it. For example, this was my undergraduate research project. Thanks to bird banding data, we get to see how house sparrow and the Eurasian tree sparrow had decreasing number along the years. This can even be published if it's a really nice project. And this one, for example, it's a case that I wanna remark. There's this festival going on through June and July at the Botanical Garden near the university. Well, in fact, this picture is taken from the building. Each night there will be different singers or bands, and of course, this will impact our birds and fauna. We have seen how some species are no longer there, or how some of them are less abundant. This was also a research project from one of my colleagues. We also have lots of nest boxes. We hang them on the trees, as you can see here, my colleague. And we visit them weekly during the breeding season. Before the nestlings go out of the nest, we will band them. And again, we will take some samples if they are needed. And after the breeding season has ended, we will be cleaning them up and collecting all the contents so that for the next breeding season, they will be free and empty and of course clean. About the wild stork colony that I was telling you before, this is not really on campus. It is on another town close to Madrid. But I think it's also a really important research that we have on, because not only because of their longevity in time, but also because it is a really nice example of this avian community. The white stork, it was endangered before the 80s at Europe. However, right now we have a really high number population in Spain, and this was all because of landfills. Once landfills started to open in the 80s, their population numbers went up. And why is this? Well, because they feed on them due to they have a food source that is constant, abundant, and predictable. Here we are finding some nestlings. And what do we actually do? Well, we go up on the nest and we place some camera traps so that we can identify each individual. Since this is a project from the 1999, and this species is philopatric, which means that they will go to breed to the same place that they were born. We have several number of individuals. We can see here their band. And once they have laid their eggs, we will measure them. We also have some project where if these eggs are not hatched, we will analyze them for some toxics, some other contaminants. And well, we check up on the hatchlings, and we will band their nestlings. Of course, again, we will take samples depending on which projects is going on. And this comes my research. I have always been interested in how human affects nature. So of course, with wild storks feeding in landfills, we can assume that they will get some impact. This is their nest. So you can see that they sometimes bring plastic bags or some other stuff as this kind of plastic. This you can see one in the actual landfill. And one curious fact, this is a bird band from Germany, so that you can see how migration works and how they actually come to Spain. And of course, my research started along this topic. So it is about how the presence and effect of environmental pollutants and plastic waste on anthropophilic bird species. So why plastic? Well, it is estimated that by 2050, there will be 1,200 million tons of plastic in the environment. This is a lot. And because of how plastic affects bird species or even wildlife. Not only physical restrictions, as they could get entangled or they could harm themselves, but also they will have some chemical alterations in case they ingest them. And another thing that I want to remark is that most research was on seabirds. So what is happening on terrestrial birds? I mean, we cannot escape out of the fact that we have this kind of rabbit mountains in nature. So my first research was on white stars and how they will feed on plastic. We will go up in the nest and we will pick up some regurgitated pellets, which I guess that you know what they are, but they are this solid material that they will regurgitate because they don't digest it, as in bones, hair, dirt, rocks, and of course, plastic. As you can see here with a rubber band or this weird element. So we will look through a stereoscopic microscope and FTIR, which stands for Fourier Transform Infrared Spectroscopy. And this is what we find, well, some part. All of these are made out of plastic, all of them are synthetic. And well, you can see here that plastic comes in different shapes, different colors, different sizes. For example, and also different polymers, as in this one is polyethylene, which is kind of like a light film. This one is also polyethylene, which is solid. This is PVC. This is also PVC. So you don't really know which kind of plastic polymer they are ingesting. So, well, this is just to show you how we analyze them with the FTIR. This will be polystyrene and how the wavelength of the polymer. So this is some results. On average, the 3.44% of the total weight of the pellet was made out of plastic. You have to have in mind that plastic is usually a light material, and we are comparing it with rocks, bones, that is some other parts that are in the pellet. So this is a really high quantity, and also because every single pellet contains plastic elements. In this graph, you can see on bars how, well, the number of pellet samples that we had 50. So that will be polyethylene in all of them, polystyrene, polypropylene in almost all of them. And the dots represents the total consumed plastic percentage. As for example, polyethylene will be almost 60%. So the most abundant polymer would be polyethylene in the total number of samples. So this is important because in literature you can see how these polymers are toxic or not. These two, polyethylene and polystyrene, some researchers here at the US, they have found that American black vulture had been ingesting those, and that there were toxicological impacts. So we have to take this into consideration, and we can just not let this slide. Some other ongoing projects that I have right now, the common swift is this bird that they will never get on the ground. They are always flying. I know that you have some other species here. So they will only stop flying when they get to lay their eggs or while they are feeding them. So any kind of plastic elements that we find, they will be atmospheric pollution. So we are at the moment doing this, well, not right now because I'm here, but I will keep on doing that. Right now, I'm working with White Ibis, here with Dr. Sonia Hernández. We are trying to compare the gastrointestinal contents from urban and natural populations, trying to relate it and to see if there are more plastic elements in urban populations or not. Of course, this species is also usually human fed, so that would be interesting to see. And it will highlight the environmental pollution that we have. And this little guy is the Eurasian tree sparrow. It is a natural species, but lately it has been becoming more urban. and they use a lot of plastic on their nest. You can see here, this was all just from one nest. And as we have been taking and collecting the nest boxes contents, we could see in time how this pattern has changed since we have some nests like for 10 years or so. We want to look at how plastic how the amount of plastic that they use have been changing on time. So this is what I have been working on and what I will be working on, and I want to thank some people. Of course, Dr. Jose Ignacio Aguirre, which is the coordinator for the vertebrate section, Dr. Francisco Cabrero, which is the coordinator for entomology, and Dr. Jose Antonio Molina, which is the director for the plants team. And well, thank you so much. Thank you for assisting and coming today. And well, we are open to any kind of collaborations that you would like to make in Spain. Thank you.
Yeah, plenty of time for questions, so who wants to start us off?
I have one. For the swifts, if you're analyzing the amount of microplastics found in the swifts, are you collecting, like, is that just fecal samples as well? Or how are you collecting that kind of data if they're constantly flying?
We work with recuperation centers, wildlife health centers. So yeah, any swift that will get into the system and they will pass or they have to be euthanized,
Yeah.
we will be collecting those.
Gotcha.
Do you ever take blood samples for like microplastic testing?
We haven't yet because trying to analyze plastic in blood samples is really difficult, as in it's really small. Well, of course, they will have a really small size. It will be nanoplastics. Well, I'm working on that with some equipment that we have. It's not going so well, so yeah, not at the moment, but it will be. It's our goal. Yeah. Thank you.
I don't know if you're the right person to ask, but when you showed the slide with how you're trying not to mow and let wildflowers grow for like the invertebrates, how do you keep like invasive plants taking over? You know?
Yeah, well, it's a really great question. I don't know if I could answer completely, but here at the university, well, there at the university, we are just letting them to grow free, and we don't have lots of invasive plants there at the campus. So that's not that's not a problem that we have at that place. I mean, we don't have lots of invasive plants at Madrid, but that's not the case of the university.
Does how long the plastics have been in the landfill affect how much is ingested or like how it breaks down inside after they eat it?
Well, the landfill is almost working all the day. So when the rabbits will get at the landfill, there are like this kind of trucks that will just like compress all the rabbits, and they will get some dirt and some land on the rabbits. So you cannot really estimate that because it's constantly they are constantly bringing more rabbits. But yeah, depending on the amount of time that the plastic is out on the nature, they will be breaking down more easily. Yeah, and that's also a problem because maybe this, I'm sure that these storks, they know what they are taking, but if they are taking some chicken breast, they cannot separate if there's some plastic piece. So yeah, that's a problem because once the plastic is out in nature, it will break down. So, yeah.
I'm not familiar with the literature, but is there a lot of are there cases of, birds having gastrointestinal health problems or causing, or gastrointestinal mortality from ingested plastics, or are they really just passing it?
Yeah, in fact, there's this study, this research, with livestock too, that they found some individual and their stomach was full of rubber bands. They might be thinking that they are worms because it's similar. So yeah, they had their whole stomach was full of rubber bands.
Did you, I think you said their population grew when y'all started having landfills?
Mhm.
Were they like threatened or anything before that? Or was
Yeah.
that, yeah,
And they were endangered. In
Okay.
fact, some the north of Europe, some populations still are endangered. The thing is that due to landfills, they have not been migrating as they should. As in, the northern populations will go down to Africa to migrate. The Spanish population will too. The Spanish population still does. They go down to, they pass the Sahara. However, the northern populations, they are staying in Spain. They have shortened their trip. So this was made out of the landfills because they don't find the need to go all down to Africa as they can feed on their landfills. So yeah, their populations, they were in danger before. I don't think they were critically in danger, but they were in danger. So in the 80s, since the landfills started to open, the population's numbers started to grow. However, in Spain, these numbers are way better than in the northern populations of Europe, due to this fact that they will migrate to Spain, but we have stocks all year round right now, which before we only had for the summer months. So yeah, they were in danger. They are not so much as right now, at least in Spain, but they are a bit in the north here.
I have two questions. The first one is the birds that are regurgitating the microplastics. Are there any signs that that's having any adverse effects on the stuff that they're?
I'm sorry, could you repeat?
Is the birds that are regurgitating the microplastics, are there any signs that those birds are having any negative effect from having in their system?
Not at the moment. We haven't looked at blood samples or some other kind of inflammation status. We have only observed that they are ingesting plastics, but we haven't studied further. We are going to in the future.
And then my second question was, are you collecting feces from those same birds to see if any microplastics are making it all the way through their system?
Yes, we have some research going on right now. We try to relate well, of course, they are alive, so we cannot get to see how their GI is. So we want to try to relate their plastic quantity in their pellets and the quantity plastic in the feces.
Did the age of the pair affect either the amount or the type of plastics used?
We didn't see any relation. They are not, yeah, I mean, there's no. What we have seen, which I didn't say, we get to know which individuals are feeding in the landfill because due to the banks that they have. But we have seen that they are getting plastics not only from the landfill but also from nature, because we have seen sometimes that even though they weren't feeding at the landfill, we have seen plastic in their pellets. So yeah, that's something remarkable too.
For the future, for the volume of blood needed, is it a really teeny amount of blood, like volume wise, to check for like nanoplastics? Or, I mean, they're so teeny
like a songbird would be so small, you're limited in the volume you can take, so like.
I don't really know. These are pretty big, so I think that we will, well, the higher volume, the better, I will guess, but we are not, we don't really know. So.
Oh, that's a lot of great questions. Thank you so much. Don't forget to, complete all the requirements for the course. Enjoy it, and this is it. See y'all later.
It's over now. I don't wanna bother you so bad. So we don't cheat. Oh, no, it's so
Yeah.
The little black. Yeah, right there.
She ain't that.
No, none of my classes.
This is, yeah.
I'm like, yeah. Thank you.
It's. Sounds like you're on top of things.